Being a successful multicellular organism requires that cells function harmoniously. No cell is allowed to make too many of its own kind or hog resources, much less invade neighboring tissues or spread to distant sites. But that's what happens in cancers. Cancer cells proliferate rapidly. They ignore biochemical signals to stop reproducing. They ignore signals telling them to die. And instead, cancer cells hijack metabolic pathways, for example, forcing the citric acid cycle to run backwards. They hear "you must divide" messages and ignore stop signs that would prevent division. They mutate genes and rearrange chromosomes. They set up their own blood flow networks and invade nearby tissues and metastasize. Instead of maintaining connections with neighbors, they spread throughout the body. In short, cancer undermines many forms of cell cooperation we take for granted in the body. In this lecture, we'll examine some of the ways that cells enter the cancer on-ramp. We'll see mutation breaking down safeguards that would ensure cells divide only when appropriate. We'll also see how research has guided development of new treatments for cancer. And you'll learn why elephants do not get cancer. There are many different kinds of cancer, but one thing all types of cancer have in common is unregulated cell division. That contrasts with good old regulated cell division, which converted us from a single-cell fertilized egg into adulthood. Regulated cell division also helps replace worn-out cells and is essential to healing from injury. That cut on your finger from last week closed up thanks to regulated cell division. Once repaired, cells promptly stop dividing thanks to regulation that tightly controls cell division. Cells monitor signals that tell them to divide. They don't proceed unless they get the green light from a relevant growth factor, such as epidermal growth factor or nerve growth factor. Growth factors bind to receptors on the cell membrane. The directive for the cell to divide passes through multiple signaling steps that make their way into the nucleus. These signals travel complicated pathways, but the result is to set in motion the process leading to cell division. Along the yellow brick road to cell division, cells check their DNA for damage before they split in two. If there is damage, they stop and fix it. If there's too much damage, the cell undergoes programmed cell death. Special parts of the cycle called checkpoints ensure each step in the cell cycle is completed properly before proceeding to the next. On top of these considerations, cells have a natural limit on the number of times they can divide. Remember, our chromosomes shrink with every DNA replication, and after a certain number of replications, they stop dividing and die. With these safeguards, how do cells manage to get off track? Well, all cancers start with a mutation in a single cell. This mutant cell begins to divide more than it normally would. It may initially produce a small abnormal clump of cells called a tumor, but that in itself does not result in cancer. Tumors may be benign or harmless at first. If additional mutations occur, cells may proliferate faster. And the more they divide, the greater the chances for mutations when their DNA is copied. The more mutations there are, the greater the dysregulation of cellular controls. As more control is lost, cancer cells spread outside of their home tissue and are said to be malignant. They can then migrate via blood or lymph to distant sites in the body where they form secondary tumors, a process called metastasis. How can these cells keep dividing indefinitely? Well, in many cancer cells, telomerase, the enzyme that extends the chromosome ends, is reactivated. Now, while telomerase is normally inactive in most adult somatic cells, it's switched on in cancer cells. And that means cancer cells go on dividing because their chromosomes never shrink enough to wind down activities. Usually it takes many mutations to make a normal cell cancerous. And this is fortunate since mutations occur in our DNA every time cells replicate. Although DNA polymerase is very accurate, mammalian cells have several billion base pairs of DNA to copy, and mistakes cannot be avoided. The more times cells undergo cell division and DNA replication, the higher the chances are that they will have accumulated enough mutations to become cancerous. Consequently, cancer risk grows with age. Though there are mechanisms for fixing replication errors, a few escaping each time can add up over the years. Why aren't there better repair mechanisms? Well, by the standards of Mother Nature, once you've lived long enough to have a chance to pass on your genes, you've served your biological purpose. So cancer is, in some ways, a side effect of planned obsolescence. But obviously, not everyone who lives a long life gets cancer. This is because development of cancer depends on factors beyond the natural pileup of mutations. Genetics matters. So do environmental factors, including exposure to chemical carcinogens, radiation, and even certain viral infections such as hepatitis B and C. Lifestyle factors play roles too, like smoking, excessive alcohol use, diet, lack of exercise, and too much sun exposure. The World Health Organization estimates that smoking is responsible for 22% of all cancer deaths worldwide. Carcinogens cause damage or mutations in DNA. We've already seen how chemical carcinogens can damage DNA, whether strand breaks or unwanted adducts to its bases. UV damage from sun exposure in tanning beds cause the formation of pyrimidine dimers, which cause deformations in the double helix. Radon in buildings is one source of radiation damage, where the tight focus of a high-energy beam tends to cause both clustered DNA damage and points of isolated damage elsewhere. All this adds to the mutational load and increases the risk of turning a cell cancerous. Any mutations that interfere with the cell's ability to correct DNA damage make it more likely other mutations will accumulate. Control of cell division can be lost due to defects in signaling pathways. Mutations can also prevent cell cycle checkpoints from functioning normally. Genes that normally suppress tumors can be inactivated. Well, let's take a look now at some of these mutations. Mutations in two major classes of genes are crucial in the development of cancer. They are grouped based on their roles in controlling cell division. Genes encoding proteins that promote cell division are called proto-oncogenes. This is a bit like calling all adults proto-criminals or all children proto-delinquents. Proto-oncogenes are genes that code for normal cell division, but through mutation can become oncogenes, which describes genes responsible for cancer. A second class of genes whose mutation is crucial in the development of cancer are called tumor suppressors. They normally encode proteins that can halt cell division, if necessary, to prevent cancerous growth. Together, these two types act as green lights or red lights, respectively, for cell division. Too much green lighting and cells will divide too much. Too little of the red light, and again, cells divide too much. More than one kind of gene encodes green light proteins for cell division. The green lights include genes for growth factors that signal cells to divide, genes for receptors of the growth factors, and genes for signaling proteins that pass on the directive to divide, like RAS. Any of these genes encoding green light proteins, such as RAS, can become a proto-oncogene, that is, a gene in the decision process for cell division. Many cancers have mutations in the gene for RAS, which encodes RAS proteins that pass on signals telling cells to divide. The most common of the RAS mutations are in positions that change amino acids 12, 13, or 61 in a RAS protein. Each of these amino acids is in the GTP binding region of the protein, and the mutations impair RAS's ability to remove a phosphate from the GTP to become GDP. If RAS can't break down the GTP, RAS gets stuck in the active form of grow, 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 even in the absence of external growth signals. So if the green lights for cell division are on when they should be off, this results in uncontrolled cell division. Different from ordinary traffic signals, there's one more thing to keep in mind. Each cell has two copies of each gene. So the green light can be thought of as having two bulbs. When a green light is supposed to be off, but one of the two bulbs is on, that still looks like a go signal, even if it's not quite as bright. So if even one copy of a proto-oncogene is active when it's supposed to be off, cells will divide when they shouldn't. You can see a mutation in one copy of a green light proto-oncogene can cause trouble. Okay, we've seen that mutations that turn on green lights for cell division in unregulated ways can give rise to uncontrolled cell division. Let's look now at genes encoding cellular red lights that stop cell division. These are called tumor suppressors. A good example is p53, a protein we discussed in the lecture about chromosome replication. p53's job is to stop the cell cycle, providing time for the cell to fix DNA damage. And if the DNA damage is too extensive to fix, p53 triggers programmed cell death. Mutations inactivating p53 remove the brakes on cell proliferation. This is like one of those murder mysteries on a mountain road where the killer sabotages the brakes on a victim's vehicle. More than half of all cancers display mutations in the gene encoding p53. In this case, we're looking at a situation where a red light signal that should prevent cell division is off when it should be on. And given the red light function of p53, let's remember the two copies of it in the cell. So we have a red light with two bulbs. If one of the bulbs burns out but the other is fine, we still have a red light that's on. The red light won't be off unless both bulbs burn out. In this case, both copies of a tumor suppressor gene must be knocked out in order for activity to be lost. The other job of p53 is triggering apoptosis, or programmed cell death, if the damage is too severe. But if the p53 in a cell is inactivated, then the cell could continue copying badly damaged DNA and proliferate, instead of either fixing the DNA or undergoing apoptosis. The continued division of cells with damaged or mutated DNA favors the progression of cancer. Now, mutations that inactivate p53 also complicate cancer treatment. Radiation and some forms of chemotherapy, for example, work by causing damage to the cancer cell's DNA in hopes of triggering apoptosis. But if cancer cells have a fully defective p53, they may not be able to undergo apoptosis. The result can be a lot of walking dead zombie cells that will not die. The key role of p53 in getting rid of cells whose DNA is past repairing is a clue to a biological mystery from the animal kingdom. Why do elephants so rarely get cancer despite living as long as we do? Well, we saw earlier that the more cell divisions there are, the greater the accumulation of errors and DNA damage. Elephants start out from a single fertilized egg, just like we do. But they have way more cells than we do, so they have way more cell divisions. Then how do elephants avoid getting cancer? The answer is in the elephant genome. Where we have just one pair of p53 genes, elephants have a whopping 20 pairs. They also have extra copies of another tumor suppressor gene called LIF6. Between them, the proteins encoded by these genes make it more likely cells with DNA damage self-destruct. By eliminating cells with DNA damage on a regular basis, most elephants can apparently dodge bullets that would otherwise have become cancer. Tumor suppressor genes also help answer another question. Why does cancer sometimes seem to run in families? It's a matter of how those genes are mutated. Remember, both copies of genes that encode tumor suppressors like p53 must be mutated in order for there to be no suppression of tumors. The probability of a mutation in both copies of the exact same gene is low, just as you don't expect lightning to hit the same place twice. Now, let's imagine that the probability of a mutation in one copy of the gene is, say, 1 in 5,000. Then the likelihood of a mutation in both copies is 1 in 5,000 times 1 in 5,000, or 1 in 25 million. For most people, it would take the accumulation of a whole lot of mutations before both copies were inactivated. But if a parent or grandparent had already acquired a mutation in a tumor suppressor gene that they passed on to you, then you would start life with only one good copy in each cell of your body. This doesn't mean you're doomed. You still have one good copy, but the likelihood you will mutate that one copy is 1 in 5,000. That means your cells are 5,000 times more likely to develop 2 mutated p53 genes than someone who started with 2 good copies. This is the reason that women with inherited mutations in the BRCA1 and BRCA2 genes have a higher risk of developing breast cancer than the general population. Both BRCA1 and BRCA2 are involved in repair of DNA damage. Carriers of mutations in BRCA1 or BRCA2 have a lifetime risk of developing breast cancer of 60 to 80% and an elevated risk of ovarian cancer. Both BRCA1 and BRCA2 repair double-strand DNA breaks by facilitating homologous recombination. BRCA1 not only helps repair DNA damage, BRCA1 also is required for triggering the p53 pathway to stop the cell cycle. It's an oncogene, but it's also a stoplight. So mutations that inactivate BRCA1 interrupt the signaling pathway that alerts p53 to stop and repair DNA damage. As a result, the cell cycle would not stop to fix the DNA damage before further cell division. BRCA1 also binds estrogen receptors. Because estrogen promotes cell division, BRCA1's binding the receptors blocks signals that would tell cells to divide. Mutations that prevent BRCA1 from binding the receptors would allow estrogen to signal cells to keep dividing. Estrogen's role in promoting the growth of certain types of breast cancers is the reason that a drug to block estrogen receptors, like tamoxifen, is used to treat breast cancers. Like BRCA1, tamoxifen can bind to the estrogen receptors and block the signals telling cancer cells to divide. Like breast cancer, colon cancer can also run in families. One kind results from mutations in genes encoding proteins responsible for correcting replication errors not dealt with by proofreading. The cancer that results from lack of functioning mismatch repair genes is called hereditary non-polyposis colon cancer, or HNPCC. People born with one mutant copy of any of these mismatch repair genes have only one good copy to rely on. Mutation of this copy in cells of the colon can speed up the buildup of other mutations that can turn cells cancerous. Individuals with mutations in their mismatch repair proteins, which have names like MSH2 and MLH1, often develop cancerous polyps as early as their 20s. Okay, so we've talked about how excessive cell division can be triggered by individual mutations in genes for green light proteins like RAS, or red light proteins like p53. And we've seen how the loss of the ability to correct DNA damage or mutations can speed up the rate at which mutations accumulate. And although mutations routinely build up with age, if repair mechanisms are defective, then mutations pile up much faster. We also saw that inheriting one mutant copy of a tumor suppressor gene raised a person's risk for cancer considerably. But most cancers don't arise from just one of these sorts of mutations, but rather from a combination of them. As the mutations accumulate, cell division becomes increasingly dysregulated. Let's see how this might play out in the development of a form of colon cancer called familial adenomatous polyposis, FAP. In this inherited cancer, individuals are born with one mutant copy of a tumor suppressor gene called APC. The first step towards developing this cancer is mutation in a cell of the colon lining that knocks out the remaining good copy of APC. With both copies of this tumor suppressor gene inactivated, the cell begins to proliferate more than it should, giving rise to a pool of mutant cells. The rapid proliferation of these mutant cells is dependent on DNA replication, and we know that replication itself results in a certain amount of mutation. The cells get a bit more rowdy and proliferate even faster if they acquire a mutation that alters the green light of the RAS protein to be active when it should not be. This increases cell division even more, leading to the formation of a small tumor. More cell division leads to more mutations, and soon another tumor suppressor gene is knocked out. Okay, things get even crazier and the small tumor grows rapidly into a big tumor. Even more cell division and even more mutation could lead to loss of the red light of p53. Wouldn't it be nice to be an elephant? You'd have a whole bunch of extra p53, but no such luck. So the brakes are gone, the tumor becomes invasive and divides even more. Runaway growth leads to accumulation of further mutations which result in the cancer metastasizing to other sites. You might be wondering whether these mutations have to happen in this particular order, and the answer is no. Once the tumor suppressor APC is inactivated, and remember, for people who are born with one mutant copy, it only takes one mutation to knock it out, the subsequent mutations can happen in any order. The main idea, though, is that multiple proto-oncogenes and tumor suppressor genes have to acquire mutations before the cancer becomes really aggressive. This could take years to happen, but you can also see that with every mutation, the cancer gets a little more aggressive, and there is a snowballing effect as cells are released from more and more constraints. Now let's take a look at some approaches to treating cancer. One way to treat cancer is to use agents that target rapidly dividing cells. Dividing cells need large amounts of DNA nucleotides to replicate their DNA. So drugs that interfere with nucleotide biosynthesis can starve these cells of the building blocks that they need. Methotrexate is one such chemotherapy drug. It works by inhibiting an enzyme needed for the synthesis of nucleotides. Methotrexate resembles the substrate of the enzyme, which is called dihydrofolate reductase. So the drug binds the active site of the enzyme as a competitive inhibitor, blocking the enzyme's activity. The downside of such treatments is that cancer cells are not the only ones in your body that are dividing. You can see, for instance, that your hair and nails grow all the time. Invisible to you, but also dividing regularly, are the cells that make up your intestinal lining and your skin, among others. All these cells are affected by treatments like methotrexate, and it is this inhibition of cell division that explains why chemotherapy can cause sudden hair loss. Since there's no way for methotrexate to tell healthy cells from sick ones, there's no way to avoid collateral damage. Devising ways to target cancer cells specifically is the holy grail of cancer drug researchers. Such treatments would rely on the differences between cancer cells and normal cells. One of those differences is that cancer cells turn on telomerase so they can keep dividing. So what could we do to stymie them? Well, inhibit telomerase. This would not be a problem for most of the rest of our cells, but it sure would hit cancer cells. Clinical trials of telomerase inhibitors show that this may be a good option. Another interesting and promising approach, which won the 2018 Nobel Prize in Medicine and has received FDA approval for some very specific treatments, is called immune checkpoint therapy. This kind of therapy relies on harnessing the patient's own immune system to attack and clear cancer cells. T cells are types of immune cells that do not normally attack a patient's own cells. Some cancer cells get eliminated, but others dodge that system. This is possible because the T cells have on their cell surface a couple of blocking proteins that must be disabled before the T cell will attack the cancer cell. Think of a safety lock that keeps a weapon from accidentally being discharged. When you have something powerful that can kill, it makes sense to keep it from doing unintentional damage to your own cells. To allow the T cells to attack the cancer cells, the safety lock must be disengaged. The blocking proteins must be disarmed. Immune checkpoint therapy uses antibodies to disarm the blocking proteins, freeing the T cells to do their work. And happily, once the safety lock proteins are disarmed, they can no longer prevent the T cells from going to town on the cancer cells. So there's still a lot to be worked out to ensure that normal cells are not also attacked by the T cells once the safety lock is disengaged. That could trigger unwanted autoimmunity similar to lupus or psoriasis. But clinical trials have shown that immune checkpoint therapies are a game changer for the treatment of several kinds of cancer, including lymphoma, melanoma, lung, and kidney cancers. The method is also being tested for other kinds of cancers and may completely alter the way that physicians approach cancer treatment. We'll look next at a couple of targeted therapies that have been developed for specific kinds of cancers based on the understanding of the molecular events involved. As we've seen before, binding of epidermal growth factor, or EGF, to its receptor triggers a signaling pathway that results in the stimulation of cell proliferation. A crucial step in the signal transduction pathway upon binding to EGF is the coming together of two receptor molecules known as dimerization. This step is essential for the divide command to be passed on. There are 4 different human EGF receptors, abbreviated HER. They're numbered 1 through 4. And while receptors HER1, HER3, and HER4 must bind EGF to dimerize, the structure of the HER2 receptor allows the receptor monomers to come together independently of EGF binding. Since epidermal growth factor isn't needed, this means that rogue dimers and the downstream effects of this signaling pathway can be triggered even in the absence of a growth signal. In normal cells, only a few HER2 receptors are expressed at the cell surface, so HER2 plays a relatively minor role in stimulating cell division. However, in about a quarter of breast cancer patients, HER2 receptors are overexpressed. Such cancers, which are called HER2 positive, can be aggressive, so therapy that blocks HER2 signaling is key to successful treatment. Antibody treatments used against HER2 positive breast cancers that have been shown effective include Herceptin and Perjeta. They work by binding specifically to the extracellular domain of the HER2 receptor and preventing it from interacting with itself or HER1, HER3, or HER4 receptors. Blocked from dimerization, HER2 cannot initiate downstream signaling. Additionally, the binding of the antibodies signals the immune system to destroy HER2 positive cells. Another example of a cancer caused by a defect in a growth factor signaling pathway is chronic myeloid leukemia, or CML. Patients with this kind of bone marrow cancer have a problem that arises from breaks of 2 of their chromosomes, 9 and 22. On each of these chromosomes, there's a gene that is snapped in 2. But that's just the start. When the cell tries to fix the damage, it ends up joining a piece of chromosome 9 to a piece of chromosome 22. And as a result, the front end of a gene called BCR from chromosome 22 gets joined to the tail of a gene called ABL on chromosome 9. When the hybrid protein is expressed, it gives rise to a mutant receptor that is always dimerized or paired up, even when there's no growth signal. Here again, rogue dimers are being formed, the same problem that we saw with the HER2 receptor we just discussed. As a result, the mutant receptor continuously signals blood cells to divide, leading to the massive proliferation of a type of white blood cells called granulocytes. What to do? Well, one way to prevent cells from dividing is to block the next step in the signaling pathway. The drug Gleevec binds to the tail of the receptors sticking into the cytoplasm, blocking downstream signaling. With no growth signal passed on, cells stop proliferating. Gleevec and its generic form imatinib has turned out to not only be a targeted therapy to successfully treat CML, but also a treatment for a range of other cancers that have similar signaling defects. In 1971, President Richard Nixon signed the National Cancer Act and created the National Cancer Institute, declaring a war on cancer. Like many conflicts, this one has lasted much longer than expected, and is one we are still fighting today. Though the enemy has turned out to be diverse and complicated, an army of researchers has designed hundreds of new weapons to push back. And there have been some dramatic successes, especially in reducing mortality for young patients. The improved survivability of children with childhood leukemia, going from only 1 in 3 to over 80%, an improvement achieved in just a few years, shows us that major battles can be won.